Cytometry, A Century of Innovation, with commentary by Dr. Louis Kamentsky. In the waking hours of the 20th century, the first fledgling steps towards what would become the art and science of cytometry began to take shape. In 1904, Coulter published photographs of epidermal cells of salamander larvae using UV illumination to show the distribution of DNA and cellular proteins.
These are really the first publications of what the DNA distribution and the protein distributions in cells may look like. So this is really the first demonstration of cytometry.
In 1924, Robert Feulgen published a technique for staining and measuring DNA, and in fact is credited with the naming of DNA. In 1936, Torbjörn Kaspersson demonstrated the ability to quantify chromosomal and cytoplasmic areas of the cell at different UV wavelengths. In the journal Acta Cytologica in 1964, Kaspersson's son showed a comparison of DNA distribution in normal cervical cells, cervical carcinoma cells, and premalignant cells, showing that cytometric measurement could be a basic tool for pathologists.
Robert Mellors, working at Cornell Medical College in New York City, has been overlooked as one of the early cytometry pioneers. In 1950, he scanned cells in the ultraviolet with a reflecting objective lens to measure extinction, and scanned cells with a cathode ray tube flying spot scanner, shown in this slide of his 1951 science paper, to quantify the fluorescence of cells. Cytometry quantification was done either using an image scanner or manually with a microscope. The latter required a way to overcome the distributional error of Beer's law. 3 different approaches to enable quantization of constituents were published by Mort Mendelsohn, Len Ornstein, and Klaus Peytot. This slide is taken from Mendelsohn's 1958 paper. To my knowledge, the first reported instrument to count cells flowing in a tube was made by Moldavan and was published in Science in 1934. Several other cell counting instruments were described by others in the 1950s and 1960s, and Parker and Horst in this patent go one step further and describe an instrument to count both red and white blood cells using 2 detectors. A 1953 development by Crosland-Taylor to more accurately optically count cells would become the sheath flow later used in all flow cytometers. Of great importance was the invention of the Coulter Counter in 1956 by Wallace Coulter to count blood cells using the change in conductivity as cells pass through an aperture.
I was at that time working at IBM on character recognition, and we had developed scanners in order to do that. IBM at that time, in '63, was very interested in getting into medicine, and a number of us, including myself, were involved in developing a strategy for IBM getting into that. And one of the areas was trying to develop a technique to screen Pap slides. That was meant to find cancer cells on stained slides. I developed a relationship with Mike Mohammed and Leo Koss, who was head of pathology at Memorial Sloan Kettering, to try to work together to develop a technique to recognize cancer cells. And obviously the first thing to do was to look at what had been done before. I spent the first few months getting samples of Pap smears from Memorial, and then I was using the Kaspersson techniques of looking at photographs of the cells at the ultraviolet at 2,600 angstroms and I think 2,700 angstroms. 2,600 angstroms gave you what they looked like, the DNA looked like, and 2,700 angstroms gave you what the proteins look like, and those photographs are actually scans of different cell populations. Then I developed a technique to scan those photographs. I think the left set of photographs are normal and cancer cells taken at 2 different wavelengths, and these are actually scans through the cells at these different wavelengths, and I show that the cancer cells look very different than the normal cells. Their scanning profiles are very different, and the amount of absorption was very different. It became very apparent back in '63, these ultraviolet measurements were interesting, but you couldn't develop a technique. Computers were then, roomfuls of buildingfuls of equipment, very slow. Lasers hadn't been invented then, so that you couldn't get enough light through a microscope to measure things very rapidly. Although these things were interesting, you had to look at something like 10 to 100,000 cells in a few minutes, and that was just out of the question with a scanning technique back in '63. I went back to thinking about physical techniques and came up with the idea of this simple apparatus where I pumped cells through a flow channel, putting it on a microscope, using an ultraviolet light source, and using a syringe pump to pump a sample through that flow chamber, and then building an assembly with 2 photomultipliers and a dichroic mirror, then measure the light loss, and then simply amplify the 2 photomultiplier signals, inverted, and then make them deflect in the standard oscilloscope X and Y displays, and then differentiate the signal and make a pulse out of it to intensify the oscilloscope display, and then you got these pictures, these dot patterns. Again, I think the invention here is correlated measurements, being able to measure 2 different constituents at the same time and produce the display like this. Later on, these dot displays were very difficult to talk about. Back then, biologists didn't know 2-dimensional things. This is a photograph of the very first flow cytometer instrument. Now you see the microscope on the right. I think it was a Bosch and Lowe microscope. A Harvard apparatus syringe pump on the left. The cells were in a coiled tubing that was on a shaker in the middle. That above the microscope was the 2 photomultipliers. Back then you used stuff that was used in nuclear physics to get pulses, and there was a rack of equipment on the right. You ended up with the display on an oscilloscope with a Polaroid camera to take pictures. There weren't memory devices back then of any kind, so your memory was a Polaroid picture. I probably ended up using more Polaroid pictures film than anybody alive back then. In '65, we developed a sorter. We showed that there were differences between cancer cells and normal cells using flow cytometry. It was far from perfect, so that it was very important to somehow find the cells that you thought were cancer and get only those cells onto a slide by themselves, so that we were very interested in cell sorting. We had looked at droplet sorting. Pap smear cells are big, and they clump together, and they just don't lend themselves to droplet sorting, so that we abandoned that technique, unfortunately. That became the standard for flow cytometry, and I actually have an early patent on droplet sorting as well, but not as elegantly as later. I'll show it Fulwyler done it. So that we developed a technique where we could actually sort cells using a fluidic switch sorting. IBM at that time had developed a typewriter using fluidic switching. Don't ask me how it worked. It was being developed in Zurich, Switzerland. So I developed a relationship with the guy who developed the fluidic switching device at the Zurich lab, and went over there and worked out this technique where we could etch that pattern you see in the center there on a molybdenum plate where the cells go through, and then if you pulse that leg that's coming down with a syringe coupled to a stepper motor, and then if we saw a cancer cell, we'd pull back on the syringe, and the cancer cells would end up in that side arm, and we could later flush the side arm out and flush it through what's called a millipore filter, and that the cancer cells would get trapped onto that filter. Then we could take the filter and then stain it as you normally would stain a pap smear, and if it worked, you would see cancer cells at that one little spot in the center. And it actually worked, and that paper was published in '67 describing it. That picture is actually showing a cancer cells. This is what they look like on the filter. It actually did work.
This slide shows our IBM instrument in 1966. It had measured 4 wavelengths of either extinction or fluorescence, had an automatic sampling system, and was interfaced to an IBM 1620 computer. 2 of the instruments were built. The second instrument was placed in Leonard Herzenberg's Stanford lab, where I collaborated with Len during the summer of 1967. Bill Bonner, working with Len Herzenberg at Stanford, developed the first fax flow cytometer in 1972. The sorting method that was applicable to blood cells and extensively used was developed by Mac Fulwyler based on Dick Sweet's ink droplet printing technique invention. Both are shown here. Wolfgang Guda developed and published in 1969 a different type of flow system which the cells were flowing through the focal plane, as shown in this paper with Dietrich, and this was later commercialized as the Feulgen instrument. In the early 70s, in addition to Herzenberg's and our development, researchers at Los Alamos contributed to flow cytometry with a number of important papers, two of which are shown in this slide. The evolution of the widely used research and clinical technique for counting lymphocyte subsets is shown in this slide. In 1971, Dick Adams and I showed that lymphocytes, monocytes, and granulocytes could be separated by measuring the fluorescence of their cytoplasmic granules. In 1975, a Los Alamos group headed by Gary Saltzman showed that these granules could be quantified by measuring forward angle scatter alone. Bob Hoffman, Patrick Kung, and Peter Hansen, working in our Ortho lab, capitalized on this to first demonstrate T-cell subsetting, as shown in their 1980 PNAS paper.
I left IBM in '68 to start a company called Biophysics Systems. Biophysics became Ortho Instruments in '76. It was bought by Johnson & Johnson.
Dr. Kamensky's company, Biophysics Systems, introduced two of the earliest cytometers as we now recognize them. The cytograph, which measured light absorption of chromatic stains as well as forward scatter, and the cytofluorograph, which measured two wavelengths of fluorescence and forward scatter. The FC200, with an improved optical design, was launched in 1975. Ortho Instruments introduced the ELT8 flow cytometer in 1977, targeted toward clinical hematology. The 50H, a research flow cytometer, followed in 1978. The Spectrum, designed for T-cell subset counting, was launched in 1979.
In the early 80s, I worked with Howard Shapiro to develop a flow cytometry method to measure T-cell activation using transmembrane potential dyes. I was interested at that time in looking at the activation of T-cells exposed to antigens as a means of detecting disease. Because distributions were so wide, flow cytometry could measure the shifts in membrane potential of the total population, but wasn't to see shifts of small subpopulations. I thought that if we could measure the shifts in fluorescence of each cell before and after antigen exposure, we might have a chance to see shifts in a subpopulation. I felt building a laser scanning cytometer in which we could measure individual cell kinetics might work, and I wanted to get back into the lab and complete what I had tried to do some 25 years earlier. So I left Johnson & Johnson and started CompuCyte with my son Lee. The development of the initial LSC instrument was done in close collaboration with a team of engineers from Olympus led by Kitagawa-san. Extensive validation studies of the prototype instruments were performed by Spizak Darjankevich at the New York Medical College, in Professor Sasaki's lab in Morioka, Japan, and by David Martin Ray in Ed Sebas's lab at the Brigham and Women's Hospital, Boston, to show the comparable performance of LSC to flow cytometry in accuracy, sensitivity, precision, and dynamic range, combined with the ability of the instrument to visualize cells of interest.
LSC technology continued to evolve, with applications ranging from drug discovery to basic research to research pathology.
Lou Kamensky was one of the earliest players in cytometry development with his early fluidic sorter and spectroscopic analyzers. The work he did before Fulwyler, Vandella, Herzenberg, or any of the important developers of cytometry hardware were successful established many of the fundamentals of cell analysis. Lou can truly be considered one of the fathers of cytometry. As president of ISAC, I am very excited that Dr. Kamensky took so much of his valuable time to narrate this presentation. I am sure that many students will find it valuable to hear directly from Dr. Kamensky as to how he came up with the many innovative ideas in the field of cytometry.
This presentation is Dr. Louis Kamensky's recollections of the history of cytometry and the part he played in that history.